Hello friends, welcome back to the series of lectures of immunology. This is the 6th lecture that will be dealing with the major histocompatibility complex, as we have already dealt in the previous lectures with the various properties of antigens as well as various properties of antibodies and their serological reaction. It will be easier for us to understand the next topic, that is major histocompatibility complex, which is also abbreviated as MHC. Major histocompatibility complex is abbreviated as MHC. MHC are cluster of genes responsible for immune response, transplantation antigens and proteins of complement system. Its products plays an important role in recognition and discriminating self and non-self. MHC was discovered by Gorer in 1930. The MHC participate in the development of both humoral and cell-mediated immune responses. In human, MHC is found on chromosome 6 and is referred to as HLA complex, whereas in mice, MHC is found on chromosome number 17 and there it is referred as H2 complex. The antigen which is responsible for graft rejection is called transplantation antigen or histocompatibility antigen. These gene segments is named as major histocompatibility complex because their discovery was based on transplantation experiments. The product of MSC is histocompatibility antigens and other antigens which are associated with immune response. MHC products are highly polymorphic. Now, there are many important aspects of MHC. Each MHC molecule has only one binding site. MHC molecules are membrane bound. Recognition by the T cell requires cell-cell contact. Cytokines and especially gamma interferons increases the level of expression of MHC. Endogenous peptides are associated with class 1 and they are recognized by cytotoxic T cells, whereas exogenous peptides are associated with class 2 MHC and are recognized by T helper cells. Now, there are three important classes of MHC: class 1, class 2 and class 3. Class 1 MHC genes, these are the glycoproteins expressed on all nucleated cells. Its major function is to represent processed antigen to cytotoxic T cell, whereas class 2 MHC genes are glycoproteins expressed on B cells and dendritic cells and other antigen presenting cells, and they play a major function to present processed antigen to helper T cells. So, here we can find a difference that class 1 deals with the cytotoxic T cell and the class 2 MHC genes, they deal with the helper T cells. The another class of MHC is the class 3 MHC genes. They form the products that include secreted proteins that have immune functions, for example, complement system, inflammatory molecules, etc. Class 1 MSC genes are found in the region of A, B and C in humans. Class 2 MHC genes are found in the region DR, DP and DQ. Class 1 and class 2 MHC share structural features. They both are involved in antigen presenting cells. Class 3 MHC have no structural similarity to class 1 and class 2, for example, heat shock proteins, complement components, etc. Now, let us have some details of class 1 MHC molecule. It is comprised of two molecules, alpha chain and beta 2 microglobulin. Alpha chain is about 45 kilodalton, which is a transmembrane chain, whereas beta 2 microglobulin is 12 kilodalton chain. These chains are non-covalently associated with each other. Association of alpha chain and beta 2 microglobulin is required for surface expression. Alpha chain is made up of three domains: alpha 1, alpha 2 and alpha 3, whereas beta 2 microglobulin is similar to alpha 3. Alpha 1 and alpha 2 forms peptide binding cleft, that is the protein they bind in this region. It fits protein of about 8 to 10 amino acid long. Alpha 3 region is highly conserved among the MHC1 molecules. It interacts with cluster of differentiation 8, that is CD8 of TC molecule, that is cytotoxic T cell molecule. This figure shows is a schematic diagram of class 1 and class 2 Mhc molecule showing the external domains, transmembrane segment and cytoplasmic tail. The peptide binding cleft is formed by the membrane distal domains in both class 1 and class 2 molecules. The membrane proximal domain possesses the basic immunoglobulin folds. Thus, class 1 and class 2 Mhc molecules are classified as members of the immunoglobulin superfamily. Talking about class 2 Mhc molecule, we can say that it is comprised of two chains, alpha and beta chain. Alpha chain and beta chain are associated non-covalently. Alpha and beta chains are made up of domains, particularly two domains, alpha 1 and alpha 2 in case of alpha chain and beta 1 and beta 2 in case of beta chain. Now, alpha 1 and beta 1 forms the antigen binding cleft. CD4 molecules binds to alpha 2 and beta 2 domains. Now, let us see how the Mhc classes they have the interaction with the peptides. Coming on in this slide, we are going to discuss about class 1 Mhc peptide interaction. Peptide presented through Mhc1 are endogenous proteins. The peptides are then transported from the cytosol into the endoplasmic reticulum. In coming into the features of peptide, the peptide is about 8 to 10 amino acid long. It interacts with class 1 Mhc molecule. As few as 100 peptide per Mhc complex can activate cytotoxic T cell. Peptides associated with the Mhc1 to cytotoxic T cells are called as target cells. Here in this diagram we can see how Mhc class 1 and Mhc class 2 have the peptide binding grooves where the peptides get associated, and here as we can see in the diagram that Mhc class 1 in this class of Mhc, only one chain, that is the alpha chain, is associated with cell membrane, whereas in Mhc class 2 we find that both the chains, that is alpha as well as beta, they are associated with cell membrane. Coming on to the interaction of class 2 Mhc and their peptide interaction, we can see here the peptide presented through Mhc2 are exogenous. They are processed through endocytic pathway. Peptides are presented to helper T cells. Most of the peptides associated with the class 2 Mhc molecules are derived from membrane bound proteins. Binding is due to the central 13 amino acid. Mhc1 fits exactly. This is not the case with the Mhc2 peptides. Now in this slide we can see the cleft geometry where the association of Mhc1 and Mhc2 can be seen with their respective peptides. Mhc class 1 accommodates peptides of 8 to 10 amino acids, whereas Mhc class 2 accommodate peptides of more than 13 amino acids. Now let us talk about the expression of Mhc class 1. Mhc class 1 is expressed via endogenous pathway due to the protein present in Mhc1 is derived from endogenous antigens. Cytosolic enzymes remove more amino acids from peptide. Peptide antigens are now transported into the endoplasmic reticulum through a pore formed by a protein called transporter associated with antigen processing, that is TAP. In endoplasmic reticulum, peptides bind with the Mhc1 by a group of proteins. Then Mhc peptide complex moves to the cell surface and integrate to the membrane. Here the complex is recognized by cytotoxic T cell. The part of antigen that binds to the Mhc is called agritope. Now let us talk about the expression of Mhc class 2. Mhc class 2 are expressed via exogenous pathway. Peptides present in T helper cells come from extracellular microbe or their products. Antigens are bind to heterodimers in the endosomal compartment rather than endoplasmic reticulum. Once peptide is bound, Mhc2 alters its conformation and trapping the peptide for presentation at the cell surface to Th cell. A invariant chain of endoplasmic reticulum ensures the binding of Mhc2 and peptides. This variant chain is cleaved by proteases. However, short segment of this chain is called CLIP, that is class 2 associated invariant chain peptides, and is remain bound and preventing any premature binding of antigen peptides. A Mhc molecule HLA-DM is present, which acts as a catalyst by removing the CLIP and promote the binding of peptide to MHC. This fragment is then released and transported to plasma membrane. In this diagram, we can see that the CD4 helper T lymphocyte can recognize peptide antigen only when presented along MHC2 molecule, whereas CD8 cytotoxic T lymphocytes can recognize peptide antigen only when presented along MHC1 molecule. Now, this is the difference between CD4 and CD8 cytotoxic T cells. Now, let us find the major differences between Mhc class 1 and class 2. When we come to the class 1, here we can say that they are present on the surface of all nucleated cell, whereas class 2 is present on the surface of antigen presenting cell. Class 1 consists of two non-identical chain, that is one long alpha and one short beta, whereas class 2 consists of the two identical chain, that is one alpha and one beta. Class 1 MHC is mainly interacts with the cytotoxic T cell, and class 2 it mainly interacts with the T helper cells. In class 1, the alpha chain is coded on the chromosome 6 and beta chain is coded on chromosome 15, whereas in Mhc class 2 both the chains are coded on the chromosome 6. In class 1 Mhc they are encoded by HLA, HLA-B, HLA-C genes, whereas Mhc class 2 are encoded by HLA-D genes. In Mhc class 1 is responsible for graft rejection and clearance of endogenous antigens, whereas Mhc class 2 is responsible for graft rejection and clearance of exogenous antigens. Coming on to the main function of MSC, MSC controls the production of antigen located at lymphocytes. MSC genes are responsible for the control of the level of complements like C1 and C2. MHC complex helps in the T cell recognition. MSC is responsible for intense graft rejection. Several non-immunological functions of MSC are like egg production in chicken, etc. These are the references that you can go for. Thank you so much for understanding MHC and their main properties. Thank you.